As we walk down a street, our brains unconsciously record details of our surroundings and the distance we have traveled to help us recall them later. Now researchers believe they have seen the building blocks of these memories as they are being formed. Scientists were able to watch the brains of mice light up as they walked on a treadmill, and as they then later rested, they found the neurons appear to fire in the same sequence while resting as they did when on the treadmill, as if the animal is mentally retracing its steps. It shows that the brain seems to encode distance in a certain way and may help to explain how we attach memories of experiences to individual places or time. Dr. Rosa Cassart, a neuroscientist at the Institut de Neurobiologie de la Méditerranée in Marseille, France, and her colleagues believe these may be the building blocks of memories as the mouse retrieves them. This sort of replay when resting is thought to be a crucial step in the formation of memories and perhaps helping the animal make decisions in the future. Speaking to Mail Online, Dr. Cassart said, "It is known that reactivations occurring during quiet rest can have a role in action planning or decision making." The researchers, whose work is published in the journal Science, added a fluorescent protein to the neurons of 5 mice so they would light up when they were active. This allowed them to monitor the activity of more than 1,000 neurons in each mouse as they walked and rested, while researchers have used implanted electrodes to monitor the activity in the brains of mice before. Using fluorescent proteins allowed the researchers to observe activity over a far larger part of the brain. When the mice were running on a treadmill, a series of neurons light up in sequential patterns. Dr. Cassart said, "The activity we recorded during running is a representation of the distance traveled in the mouse hippocampus. When the animals were resting, however, they saw the same cells light up but in a much faster way. These seemed to persist even for several days after they had been on the treadmill. They also appeared to light up in sequential blocks that corresponded to particular fragments of the run, almost like they were fast-forwarding through their journeys, recalling chunks of their time on the treadmill at a time. This may also help the neurons to strengthen or weaken their connections over time depending on how important the memory is, a process known as synaptic plasticity." Dr. Cassart added, "The replay is thought to occur faster to allow for synaptic plasticity to occur between the neurons involved." Surprisingly, the neurons for each piece of the memory were also mixed up throughout the hippocampus rather than being right next to each other. Instead, they seem to fire in distinct patterns, triggering neurons in other parts of the brain as it recorded the time and distance on the treadmill. However, the animals were not given any other sensory input, such as changing scenery, smells or sounds, like they would get in the real world. This would suggest the memories formed then may be a lot more complex as these senses get mixed in with different distances. Cameron Duba, a neuroscientist at the University of Wisconsin in Milwaukee, told New Scientist that it was surprising to see an experience broken down into segments during recall. He said, "The cells essentially fire in order throughout the run, so why would it break down into discrete assemblies?"